In the vast universe of knowledge, few discoveries have been as profound as deciphering the code of life. DNA. This molecule, a double helix resembling a twisted ladder, holds the blueprint for every living organism on Earth. Understanding this code, reading the sequence of its building blocks has revolutionized biology and medicine. Imagine a vast library filled with books written in an unknown language. Each book represents the DNA of a different species, and within its pages lie the instructions for life. DNA sequencing is the key to unlocking these secrets, providing us with the tools to read and understand the language of life itself. From understanding our own ancestry to developing personalized medicine, DNA sequencing has become an indispensable tool in the 21st century. The journey into the heart of DNA is a testament to human ingenuity and our insatiable thirst for knowledge. Before we can understand how DNA sequencing works, we must first appreciate the elegant simplicity of DNA structure. Imagine a ladder twisted into a spiral. That's the iconic double helix of DNA. The rungs of this ladder are made up of 4 chemical building blocks or bases: adenine A, thymine T, guanine G, and cytosine C. These bases pair up in a specific manner, A always with T and G always with C, forming the genetic alphabet. Like letters forming words and sentences, the sequence of these bases along the DNA strand carries genetic information. This sequence dictates the production of proteins, the workhorses of the cell, which carry out a myriad of functions essential for life. Deciphering this code, determining the precise order of these bases, is the essence of DNA sequencing. By deciphering this code, we gain access to a treasure trove of information, enabling us to unravel the mysteries of evolution, disease, and the very nature of life itself. The first breakthrough in DNA sequencing came in the 1970s with the development of Sanger sequencing, named after its inventor, Frederick Sanger. This method, often referred to as first-generation sequencing, revolutionized molecular biology and earned Sanger his second Nobel Prize in chemistry. Imagine a marathon where runners are randomly stopped at different points along the racecourse. By analyzing the positions where the runners stopped, we can deduce the order in which they ran. Sanger sequencing works in a similar way, using special molecules called dideoxynucleotides to halt DNA replication at specific points. By using fluorescently labeled dideoxynucleotides, each corresponding to a specific base, A, T, G, or C, scientists can determine the order of bases in a DNA sequence. The resulting fragments, each ending in a fluorescently labeled dideoxynucleotide, are then separated by size using a technique called electrophoresis. By reading the order of these colored bands, scientists can reconstruct the original DNA sequence. While groundbreaking, Sanger sequencing had its limitations. It was relatively slow and expensive, making it impractical for large-scale projects like sequencing entire genomes. The need for faster, more cost-effective sequencing methods led to the development of next-generation sequencing (NGS) technologies. These high-throughput methods revolutionized genomics, enabling scientists to sequence DNA at unprecedented speeds and scales. Unlike Sanger sequencing, which relies on separating DNA fragments by size, NGS platforms employ various strategies to sequence millions or even billions of DNA fragments in parallel. This dramatic increase in speed and affordability has democratized DNA sequencing, making it accessible to a wider range of researchers and fueling a revolution in fields like medicine, agriculture, and environmental science. The ability to generate vast amounts of genomic data has opened up new avenues for understanding complex biological processes and addressing some of the world's most pressing challenges. Among the various NGS platforms, Illumina sequencing has emerged as the dominant technology, revolutionizing genomics and enabling a wide range of applications. This method, based on sequencing by synthesis, combines elegant chemistry with sophisticated imaging techniques to decipher the code of life with remarkable accuracy and efficiency. Imagine a vast dance floor illuminated by flashing lights, each flash representing the addition of a single DNA base. That's the essence of Illumina sequencing, where millions of DNA fragments are attached to a solid surface and sequenced simultaneously using fluorescently labeled nucleotides. As fluorescently labeled nucleotides flow over the flow cell, they bind to their complementary bases on the template DNA strands, emitting a flash of light upon incorporation. These light signals, captured by a sensitive camera, reveal the order of nucleotides being added, allowing scientists to reconstruct the original DNA sequence. The massive parallelism of this approach enables the sequencing of billions of DNA fragments simultaneously, generating vast amounts of data in a single run. Genome sequencing determines the complete DNA sequence of an organism. Once a Herculean task, it's now routine thanks to NGS technologies. Imagine accessing the complete blueprint of an organism. Genome sequencing reveals genetic secrets, enabling detailed study of gene function and disease susceptibility. Sequencing a human genome now takes days and costs a few $1,000s. This revolution in genomics has led to sequencing thousands of genomes, including extinct creatures. It's an indispensable tool for understanding life's diversity and evolutionary relationships. Perhaps the most profound impact of DNA sequencing has been in the field of medicine, where it has revolutionized our understanding of genetic diseases and paved the way for personalized medicine. By comparing the genomes of healthy individuals with those affected by disease, scientists can identify genetic variants associated with disease susceptibility. Imagine having a roadmap of the human genome, highlighting genes involved in various diseases. That's the power of genome-wide association studies, or GWAS, which have identified thousands of genetic variants linked to complex diseases like cancer, heart disease, and diabetes. By pinpointing these disease-associated variants, scientists can gain insights into the underlying mechanisms of disease and develop targeted therapies. Moreover, DNA sequencing plays a crucial role in diagnosing and managing genetic disorders, enabling accurate diagnosis and personalized treatment plans. DNA sequencing is not just a tool for understanding the present and future, it also provides a powerful lens into the past, enabling us to unravel the mysteries of ancient civilizations and trace the migratory patterns of our ancestors. By analyzing ancient DNA extracted from fossils and archaeological remains, scientists can reconstruct the history of life on Earth and gain insights into human evolution. Imagine extracting DNA from a 40,000-year-old Neanderthal bone and comparing it to the DNA of modern humans. That's precisely what scientists have done, revealing that Neanderthals interbred with our ancestors, leaving a subtle genetic imprint on modern human populations. Ancient DNA analysis has revolutionized archaeology, providing unprecedented insights into the lives of our ancestors, their diets, diseases, and migratory patterns. By comparing the genomes of ancient populations from different regions and time periods, scientists can trace the movement of people across continents and reconstruct the history of human migration. In the realm of criminal justice, DNA sequencing has emerged as a powerful tool, providing irrefutable evidence to convict the guilty and exonerate the innocent. DNA profiling, a technique that analyzes specific regions of DNA that vary between individuals, has become the gold standard in forensic science. Imagine a microscopic barcode unique to each individual, present in every cell of their body. That's essentially what DNA profiling analyzes, comparing DNA samples collected from crime scenes to those of suspects or victims. The high sensitivity and specificity of DNA profiling make it an incredibly powerful tool for linking suspects to crime scenes, identifying victims, and resolving paternity disputes. The use of DNA evidence has revolutionized forensic science, leading to the resolution of countless cold cases and the exoneration of wrongfully convicted individuals. As DNA sequencing technologies continue to advance, becoming faster, cheaper, and more accessible, we are entering an era of personalized medicine, where medical treatments are tailored to an individual's unique genetic makeup. This paradigm shift in healthcare promises to revolutionize disease prevention, diagnosis, and treatment. Imagine a future where your doctor can analyze your genome and predict your risk of developing certain diseases, allowing for early intervention and preventive measures. This is the promise of personalized medicine, where medical decisions are guided by an individual's genetic information, leading to more effective treatments and better health outcomes. By understanding an individual's genetic predispositions, clinicians can tailor treatments to their specific needs, minimizing adverse reactions and maximizing therapeutic efficacy. Moreover, DNA sequencing is paving the way for gene therapy, a revolutionary approach that involves correcting defective genes to treat genetic disorders. The journey into the heart of DNA, made possible by DNA sequencing, has been one of the most remarkable scientific adventures of our time. From deciphering the code of life to revolutionizing medicine, DNA sequencing has transformed our understanding of the world. Each discovery pushes the boundaries of our knowledge, revealing the intricate complexities of life. The ability to read and understand DNA raises profound ethical questions about our future. The journey into the heart of DNA is far from over, it has only just begun.